dementia is not inevitable. We will all suffer a certain amount of cognitive decline with time, with age. And we have an aging population, so more of us are going to deal with this failing cognitive decline. But as mine is failing at the moment. But dementia is very different. Dementia is the death or loss of structures in the brain that then lead to the loss of particular behaviors. And I'm going to refer to the hippocampus that Lisa has just introduced to us as this structure that consolidates early memory. And that is lost, atrophies, it dies in Alzheimer's disease, and with it the loss of ability to form memory. We have an aging population, but I can say that within this room, the bad news is 30% of you will suffer from dementia at some point in your life. But the good news is that 60% of you will not suffer from dementia. So that's telling us that it's a disease. You can go through life and not have dementia. It is a disease that I insist we can identify, understand, and start to tackle as a disease. The eye-watering statistics for dementia in this country are that it's costing the economy $24 billion per year. Now, individually, as we look at individuals in the population, only 48% of the people who live with dementia receive a formal diagnosis. And that's mostly because dementia, we can't really do anything about it at this moment in time. There's very little therapy available. So it's an end-of-life condition, so what do? I mean, we say it's dementia, but we don't send the people for an expensive CT scan. But 36% of the population now will know individuals, friends or family members with dementia. So there's nearly 200 of you in this room, so that's about 70 of you. Would that be, give us a nod if you know people? Yeah, that looks about the right number of figures. But the actual costs here in this 24 billion are that 1 in 4 hospital beds are occupied by people with dementia. And that's an added difficulty with dementia is that when people with dementia are ill, they can't tell you what's wrong with them. So they have to be hospitalized till other conditions can be sorted out. They don't just suffer from dementia. 1 in 3 of the over 65s will die with some form of dementia, as I mentioned. But 2 in 3 of the care home residents at this moment in time have some form of dementia. So what are we doing about dementia? How are we researching dementia, and what's being brought to this task? Well, I'm going to make a parallel with cancer. Cancer was once considered as a disease of aging, and it wasn't really studied in any great detail. But cancer research, back a bit, cancer research receives 10 times as much funding as dementia research. Now, this disparity, the roots of this disparity are political, to some degree, historical and vocabulary, linguistic. And to bring these together, I mean, Richard Nixon in 1971 made this national commitment for the conquest of cancer, and later it was declared as the war against cancer. So we had this vocabulary and this political will coming together to deal with cancer. And we don't really have that same vocabulary for dementia. You know, we talk about people suffering from dementia, well, they're getting a bit old, he's losing his marbles, going a bit soft in the head. We don't battle cancer, we don't battle dementia, we don't fight dementia in the same way. And I think we need that vocabulary to actually bring along the political will as well. And we need that for this aging population. This is distinctly helped by the recent pledge for dementia by David Cameron in February of 2015. So what are we doing to try to understand dementia? Well, I'm working on one particular form of dementia to try to understand the early events that may actually initiate the progression. Now, I told you at the start that dementia is not inevitable, but for some individuals in the population, dementia is inevitable. And that's those individuals who have a genetic form of the disease, so it's carried through their family. And one of these diseases is, has a large genetic component, is frontotemporal dementia. And frontotemporal dementia is an early onset dementia, so it comes on in the 5th and 6th decade of life, and usually it kills the individual within 5 to 8 years of onset. Now, it deals with, it kills these structures, the frontal lobe and the temporal lobe, and these are structures that are intimately involved in social calculation and interaction and linguistic calculation and interaction. And as Lisa also sort of mentioned, vocabulary is really critical to who we are. So frontotemporal dementia is very clearly a loss of self. It's the second most prevalent early onset dementia, and it's associated, as I say, with this death of the frontal and temporal lobes. So how do we actually try to understand frontotemporal dementia? Oh, sorry, come back a second. Recently, there's been this understanding of frontotemporal dementia that it actually is a spectrum of disease. It has a spectrum with motor neuron disease. So what happens, motor neuron disease, the disease that Stephen Hawking has, is the loss of movement, the loss of muscular function in the individual. And if we look at individuals with either FTD or motor neuron disease, often we see a comorbidity, which tells that is that they have, if they have motor neuron disease, later on in the disease we see an aspect of frontotemporal dementia. And the same with frontotemporal dementia. If we look at those individuals, they'll often later in the disease have an aspect of motor neuron disease. But we also have within the same families siblings who will develop full frontotemporal dementia, and then the other sibling will develop full motor neuron disease. So what it's telling us is that this is a whole spectrum. This area is it's all one disease, most likely. There are very pure forms of the disease, motor neuron disease and frontotemporal dementia, and mutations, familial mutations in tau or progranulin will give a very pure form of frontotemporal dementia, while mutations in SOD1 will give motor neuron disease. So I've been working with this particular gene, CHIMP2B, and how do we try to understand or get to the bottom of frontotemporal dementia? Well, I'm using, strangely enough, fruit flies. So we use fruit flies because they have this very simple genetics and a very complex experimental palette that we can use. So the genetics is a very powerful tool to be able to answer some of the questions that we want to ask. And in particular, we use this fly eye. As you can see here, there's this beautiful crystalline structure of the fly eye. So any disruptions to that we can see. And so what we do is we activate the CHIMP2B, the mutant CHIMP2B protein in the fly eye, and we see what happens to that. And what we see, first of all, just to look at the normal, the wild type, here's the crystalline structure of the fly eye. And here, if we peel off this eye, which sounds a bit painful, I know, but if we look here with a microscope, if we label up structures, we can see these are the light-sensitive structures of the fly eye, and around them in green is the cell, but we've labeled up another structure that's involved with self-eating of the cell, which is about how the cell renews itself on a daily basis. When we activate CHIMP2B, what we see is this glazing of the eye. It's very clearly disrupted, and we see these black spots. And what we see is that the photoreceptors, the light-sensitive receptors, are really quite blown away, and the autophagy is very much activated and all over the place, really. So what we want to do with this is we then use this model to actually ask what advances the disease and what prevents the disease. So we're looking for canaries in the mine here. We're looking to see what are the first events during the onset of the condition. And to do this, we actually want to narrow down just on individual neurons. And we use, back to Drosophila again, Drosophila maggots. So you maybe didn't think you'd be looking at maggots today, but we are. And what we do is we think of the maggot as a tube of muscles. When we cut the maggot down the middle and lay it flat, what we see is this very nice array of muscles. Now, every muscle needs a nerve to stimulate it, and it's those nerves that we're going to focus on. And actually, these nerves down in these muscles have more similarities to the sort of nerves that are functioning in our central brain. They use an amino acid called glutamate for the signaling between cells. So this is what we see. You'll not really think about maggots in the same way again, hopefully. But this is a synapse. So this is the contact between the brain and the muscle. And they're normally in a registered size. They grow in concert with the muscle to a particular size. So when we were doing our screen for canaries in the mind to look for events that were happening before to advance the condition, we found a mutant called RAB8. And so what we did was we went down to this synapse, this connection with the muscle, to see what was going wrong when we take away RAB8. And what we found with the RAB8 mutant, when we take away RAB8 altogether, is that the synapse is massively overgrown. And what we do is we count these little structures, these pearls on a string structures are called boutons, and this is the quantification here. The synapse is nearly double in size. And what's that telling us? It's telling us that the growth control is very aberrant. So there's something going wrong between the conversation that the nerve and muscle have to keep themselves in register in terms of their size. So how do we think about that? These signals are passing between the nerve and the muscle all the time, and the muscle particularly sends a signal to the nerve, and it's secreted, and the nerve takes it up in these blebs of membrane, which are rather like a lava lamp. And so the blebs of membrane move through the cell, and one of two things usually happens. The signal is either degraded or it's recycled. And so, you know, they're very virtuous, our cells, in terms of recycling and degradation. So this is where the CHIMP2B protein normally works, and RAB8 normally works in this recycling step. So we wanted to ask if something was wrong with the growth control. So we looked at a particular signal that the two cells exchange. That's tissue growth factor beta. And if we look back in the brain, because this is where the signal is interpreted, we can see here's the motor neurons, so the neurons that are stimulating the muscle. And these are the nuclei where the signals are perceived. And this is the signal here for a wild type, and it's just diffuse, a few spots around the cell. But in RAB8, we can see that there's far, far more of it. So there's much, much more of this tissue growth factor beta signal. There's another signal that needs to work with the tissue growth factor beta signal, and that's JNK-AP1. And we do the same thing. We look back in the brain at the motor neurons, and here's the signal in the wild type, the normal larval brain. And here's the RAB8. We can see that there's much, much more of this signal. So the growth signal is clearly perturbed. How do we think about this? What's going wrong? So, you know, the growth signal's coming in on the membrane into these membrane blebs and trafficking through the cell to either be recycled or to be degraded. With some other clues that we had, we latched onto a protein that is called POSH, which stands for not port out, starboard home, for those that know that acronym. It's plenty of SH3s, and that is a bit of alphanumeric self-abuse that us biologists inflict upon ourselves. But it tells us that the protein is a scaffold. And what it's a scaffold for is this junk signal that we talked about. So it's activating this junk signal. But we know that it's normally also degrading the tissue growth factor beta signal. The thing about POSH is it's normally, you just don't see it in the cell at all. But when it's activated, it becomes abundant. And so then when it's activated, it's making this scaffold and blocking this normal traffic. So we wanted to ask, as the Spice Girls did in 1998, is POSH a problem? So we look at the synapse of a wild type, and there's the POSH mutant. So we've taken away POSH altogether here. It can't be activated. And we've got the RAB8 massively overgrown. But if we make a combination of these two mutants, so take away both proteins, we find that we revert the growth back to wild type. So POSH is a problem, and we've rectified this condition. So if we remove this POSH from the overgrown neurons, we rectify growth. And there's a couple of things to that. As I mentioned, that POSH is not normally there in any sort of quantity. So POSH is a marker, and it's also something that's causing us a problem. So it's something that we can target potentially in the future. We wanted to go back, that's a Drosophila maggot. Great, yeah. I'm standing in front of here trying to tell you that Drosophila is like a human. It is in large part. We went back to rat neurons, and we looked at POSH at the same time when we had activated this poison chimp2b, the mutant chimp2b protein. And we find that when you see this magenta and the green in the same position, that's telling you that POSH and the chimp2b are in the same position. And so you can see here that the magenta and green make white together. So where you see white is POSH and chimp in the same place. And we know that POSH actually sits on accumulations of the chimp protein now. So we think that POSH is a canary in the mine, and we're starting to see that POSH may actually be relevant to some of the other forms of this frontotemporal dementia and motor neuron disease. So we're thinking that this can help, at least at this very early stage, to identify these targets that we can later think about for tackling or preventing or slowing down dementia in the long term. And this should, in time, although frontotemporal dementia is a smaller percentage of the dementia population in general, these are the sorts of things that we need to be doing to think about this, these 850,000 people in the UK that are currently living with dementia, and providing help and hope for the future for the 706,000 family carers of people with dementia. And with that, I'd just like to thank the member. I mean, I'm showing all this work from the other people in my lab, and this is them and the funding that I received from the Alzheimer's Society, the Alzheimer's Research UK, and the Medical Research Council. And thank you.